And then once this green cell, the linker cell, reaches the tail of the animal, it dies there. And it has to die there because if it doesn't die, it kind of clogs this connection that the gonad makes with the cloaca, which allows the sperm of the worm to release. So essentially, it will result in the animals being infertile and sterile. So essentially, this is the second big job of this linker, green linker cell, that it has to die. And the way it dies, it's really interesting. And probably, you know, you read the primer, the preview, the review article. And there's a lot of complicated information, but I hope you got from it that there are different types of cell death. And there was a little section on linker cell death there. We will not go into all the details, but I just wanted to share with you one observation that really struck me when I started to study this. So when the cell migrates in the young worm, it has a very nice and round nucleus. So it's a migrating linker cell here. But when it starts to die, what we see is that this nucleus becomes quite deformed, and we call it nuclear deformations. And so I was very curious to understand how does this transformation happen from this nice and round nucleus, like in all the normal cells, to this very deformed nucleus. And why is that important? Actually, this deformed kind of nucleus is observed in all of the aggressive cancer types. And the more aggressive the cancer is, the more deformed is the nucleus. Also, it's observed in some of the neurodegenerative diseases, in the aging diseases, and diseases of the heart, is cardiomyopathies, in muscular dystrophies. So it seems to be relevant to many, many different conditions. And I'm just really, really interested to understand how does this happen. And today with you, we will tackle this first question to understand, to sort of form an observation about this. And so what I wanted to do is to look in live animals and look specifically at the nucleus using some of the cell biology tools to see how this change happens. And when we step back for a second, I will just show you a schematic of the nucleus structure. So this is the whole cell that you have. And this is the nucleus inside, and you have a DNA in here. So the nucleus has a double membrane, and we call it the nuclear envelope. And then there is this lamina in blue, which is composed of lamin protein. And essentially what I decided to do is to look at this lamin protein because it will allow me to see how the nucleus looks. And how do we do that? So that we do thanks to this awesome technology of the green fluorescent protein called GFP. This protein was discovered in jellyfish, and there's actually a Nobel Prize was awarded in 2008 in chemistry to 3 scientists who worked with this. And they saw that this protein emits green light when you shine UV light on it. And they were able to attach it to other proteins and cells. It's kind of like a piggyback. And so you can follow other proteins and see what they do in the cell or in the animal. So here, what I have is I have my lamin protein and I have the GFP attached to it. So by looking at the GFP, it's almost like I'm looking at lamin. I will know where a lamin is because lamin is invisible normally. So now when you look at your nucleus, so each molecule of lamin in the nucleus now will have this little tag of GFP, and all of them will be fluorescing when I put them under the fluorescent microscope. And so you will see the light shining, you will be able to see this nuclear shape. Okay, so I think I have to quickly move now to the my first microscopy room. I will walk you through the lab. So come along with me. We'll just see a little bit. And I have to put on my mask because now I'm entering the space where other people are working. So this is, I guess I will show you guys some of the lab as we walk through it. But we have these incubators in which we grow the C. elegans. And now we will enter the microscopy room. So it's usually dark in here. Okay. I will shut off the light, and what I will do is when you look at the worms channel now, I will turn on the fluorescence. You will be able to see something happening. So do you all see it? Put the thumbs up if you see that the worms are fluorescing. You see these tiny little green specks. Essentially, we see all of these nuclei in all of the cells of the animal. So and it's quite amazing that we can see it live here. And this is great that we can see it live. However, this is not a good enough resolution. And we want to look at the single cell, right? The linker cell and what happens to its nucleus. So we really have to go to really, really zoom in. And to do that, we will transfer to a more complex microscope, more complex fluorescent microscope to see that. So I'm just gonna, just bear with me for a second. Just need to remove my phone, my computer. We're gonna walk to a different microscopy room now. Again, let me turn so you can see the lab a little bit. We are all working in masks, socially distanced. Okay. Now this is a much bigger microscope. It has a computer connected to it, so we will be able to see images on this. Just give me a second to connect my phone. I have this adapter that connects to my phone and goes into the microscope so that all of you can see this. Okay. We'll turn on the lights. Okay, here we go. So now, essentially, to be able to see the animals so close by, you see that they are not moving here, right? I put them on these glass slides and I put a cover slip on top of them. And I added a drop of anesthesia to them. So they are essentially sleeping and they stop moving, because when I want to take a picture of the single cell, I really can't have them move because then my picture will not come out really well. So I need them to stop moving and sleep. And that's how I can take a picture of them. They can be revived after this if necessary, but usually we don't revive them because we just grow so many of them and we just can always get more.
Oh yeah, if they die, will they stop fluorescing? A student is asking.
Actually, old animals fluoresce more. They have this green autofluorescence. And sometimes you can see a dead animal, it has this autofluorescence to it. Also because they eat bacteria and their intestine is like a huge part of their body. Sometimes you can see a lot of extra fluorescence coming from there. I was just going to say that please interrupt me with questions. I forgot to say this in the beginning, but I'm happy to answer at any point. Yeah, so here essentially we see a close-up of the animal, and you guys can see, unfortunately I can't really show you with anything, but you can see the outline of one animal. Right in the middle of the animal, we have a tail, and this is where the dying linker cell will be. So now let us take a picture. Let me show you how we do it. So here we have a computer, we have special software where we have to select different channels that we want to do. It's probably a little hard to see. Now I have to unfortunately remove the filter because now all the light goes to the objective and goes down to the slide, and the output goes to my phone. However, in order to take a picture, I need to direct the output to the camera that is up here. So to do that, I need to switch. So you won't see the worms on your screen anymore, but we will take a picture. You can probably hear the noise a little bit. This is our picture. So I don't know how well you can see it from here, but let's remove some filters. So in green, you can see many green spots. So these are all the nuclei and all the cells. And then you can see this red signal. So actually this GFP protein can be modified to emit different colors. So now it will be called red fluorescent protein, yellow fluorescent protein, and other kinds. So here what I have a special red fluorescent protein that only marks the linker cell. So you can see it here. I know where the linker cell is. And by doing so, I will know which nucleus belongs to the linker cell. So I can overlap and I can see, okay, here's the nucleus that is inside this cell. And when I remove the cell, I can see the nucleus inside. And so essentially after this, this is not even the most complicated microscope that I use, but this is the best one I can demonstrate to you in the lab in this class. I actually have to go. We have this imaging facility that has probably 20 to 30 different very expensive microscopes, and I'm trained on some of them to take images. So I will show you the output of those images. I will share my presentation and just show you a couple of very short videos of what happens. Just give me a second, please. I'll share my screen with you. Okay, can people see my screen? Okay. Okay, so essentially this is from this light field microscope. And this allows me to look at the cells in 3D. And I make this these short videos. So what you have here is you have the animal in which the linker cell is migrating. It's marked in red, as we have just seen. And all of the nuclei and other cells are marked in green. So when I play this video, you can see in 3D that this is the nucleus of the linker cell, and you can see the other nuclei around it. And by looking at this, you can immediately tell me, oh, there's more green signal in this cell. And I will say, yes, I agree with you. And this is already an interesting observation. So essentially, as scientists, what we do and what I will do with this is I will take many, many pictures, like 50 to 100 pictures of worms of these linker cells. And I will look at these images and try to find the trend and try to find some observation. What do I see? What is different in the linker cell compared to other cells? what is different in this migrating cell versus when the cell starts to die. And here I have one video of the cell that is dying. So you can see the cell here, the nucleus, and actually by these images, I observed that this lamin forms this big blob in the dying cells. And as of now, I don't really know what's the significance of this, but this is the observation that I made. And now, as scientists, we have to build different hypotheses to try to answer why do we think this happens? We might want to try to perturb this lamin in the linker cell and see if things go differently, if the cell cannot survive or if it dies faster and things like that. So yeah, this is basically, I guess, the end of my demonstration. And I would be happy to take questions from you. I can go back to the room where I can uncover my face again.
So while she's walking back, y'all can think of your questions. Feel free to raise your hand and unmute yourselves, or feel free to ask questions in the chat.
Yeah, sorry, I'm here. I just
Oh, there you go. Awesome.
Everybody froze.
Yeah.
My Zoom froze. But hopefully you guys all had the chance to think of some questions that you want to ask me, and we can have a bit of a Q&A before this class ends.
So there are questions on how they make this technology. I guess they're talking about like the microscopes.
Are you talking about the microscopes, or are you talking about how you engineer these proteins that are tagged with this fluorescent tag, with this GFP tag?
Evan, would you like to clarify?
Well, like in general, because like the quality of like the things we got shown were like really good. I've not seen before, to be honest. I'm just wondering like how they even like managed to do this.
Well, the microscopes are just getting better and better. Yeah, so I mean, yeah, really, I guess there are two technologies that are used here. One that I described, this GFP tagging, right? So that is actually done by, you can now with I don't know if you guys have heard this CRISPR technology for genome engineering. So you can take the sequence of this GFP tag and attach it to the sequence of your favorite protein, your favorite gene, and you will essentially make it. So this is one technology. The other technology is microscopy, and there are so many different microscopes that you can use for this. I showed you today the camera compound with which we took a picture. I showed you the videos that I took with the wide field microscope. So it takes many tiny sections that are separated from each other, and then I use the software that puts it all together. And so at the end, you get this output of a 3D video that you can, that you can see. You can go into individual slices and you can take a look. There are other microscopes that are called confocal. I'm not a microscopy specialist, but I showed you a little bit about how, you have your slide on there and how the light goes through. Essentially, the technology is that you shine the light in the fluorescent microscope, and your fluorescent protein emits the light back that is then detected, and you can see it. And of course, the better resolution cameras we have now, the better pictures you have. So the cameras are constantly improving, and this is also another reason why we can get better and better pictures.
There was a question before about the magnification that you used for the first microscope.
The first microscope, it was only a 2x magnification. So you can, when you look at the worms on the plates against some light, you will see tiny animals. But when you want to see a little bit, usually when we want to see a bit better, you know, we use 1.6 or 2x magnification to look at the live animals. But then when you want to take images, usually at least 40x, we looked at 40x, we took a picture with 40x magnification, and I also do a 63x, and you can do a 100x to really see clearly.
Can you elaborate on why the linker cells glow brighter?
Well, that's a kind of very general tag that we use, and it's a kind of tool just to know where that linker cell is. The worm is transparent, so we can actually see it without the fluorescence, but it's better to have the fluorescent tag. It depends on what kind of tag we use. Some of them are brighter and some of them are dimmer. I have different kinds of tags for the linker cell, and I have many of the engineered worm strains that will glow different colors. So I have linker cells that can be very, very dark red. The red one that you saw, I have the ones that can be green. And so I can mix and match them with the proteins that I want to see, because sometimes my proteins I tagged with the red fluorescent protein. So then I need to take a green linker cell. Yeah.
And then there's a question on how do they differ between the linker cells and, oh, never mind. They said that answered their question.
Okay.
I guess, from your observation for when you got like the grouping of all the GFP cells, do you have any like hypotheses as to why that's happening?
Well, when I look, I definitely see that in the linker cell, there's a stronger green GFP signal, as you saw from the picture that we took together. Sorry, from the picture that I showed you at the end. And I think it's probably, it's a property of the migrating cells. They have to be a bit more flexible and be able to mold, because they move through the other tissues and cells in the worms. So the lamin allows the nucleus to be a little bit more flexible. But then once the cell is dying and you form this kind of patch on one side of the cell, that I also observed in very old worms. So in their cells, this happens as well. It happens in this aging disease called progeria. I don't know if you guys heard about it, but it's a disease when children become old really, really fast. So the same thing happens in there. And what I'm trying to understand now is, why does it happen and what genes control this? But I have many different hypotheses that I'm testing right now. And maybe in the future, I will be able to tell you about my discovery of this.
Is there a definite way to measure fluorescence?
Yes, there is a way to measure fluorescence, but in my hands, I think you have to be careful with measuring fluorescence, the amount of fluorescence. You will need to have some kind of other tag in your worm, some other fluorescent tag that you will know should be something constant, that should not change from worm to worm too much. This will be kind of like your background level. And if you want to measure any differences in what you're looking for, you will be comparing that to this kind of background level to see that the variation doesn't come just because you're looking at different animals. They are slightly different age, maybe on different slides. There is a different fluorescence, the way that you prepare the slides. So for me, I prefer not to measure fluorescence directly. I prefer to, when I look at these structures and I see this one GFP blob, I will say, oh, in this cell, I observe this one blob, and I will see, do I have any cells that have 2 blobs? So then I will count the cells. I have 10 cells with 1 blob, and I have 2 cells with 2 blobs. And this is the kind of quantification I prefer to do myself. But people do measure fluorescence. You just have to be more careful how you do it. So you don't introduce any error into your quantification.
Yeah, so it's measured relatively rather than on a universal scale. That's what they call it.
Mm-hmm.
And what does a normal day in the lab look like?
Well, right now, my main responsibility in the lab is really just research. And so when I get in, I usually have different times booked on different microscopes. So we have this booking system, so all members of the lab can plan ahead, and they will approximate how much time they will need on all the microscopes. And so we have this lab Google Calendar for different microscopes, and usually I will I also make these genetic crosses between different animals, so I need to use these small microscopes that I showed you in the beginning to determine determine what's the outcome of the cross. I usually take images, and essentially my all days are really filled with experiments now. Sometimes you need to make a conscious decision in the time that I need to go and get water, I need a break to eat, because sometimes you're just so carried by the experiment that you forget to do those things. And it also depends on how your experiments are. Are they really time sensitive? Do you have to put on a timer and you have some kind of chemical reaction running and you really have to stop it in time? And those experiments tend to be a little bit more stressful. Or for example, when I need to revive the worm, I take back that slide and I have to be very, very gentle and gently pick it off from that slide and put it on the bacteria food and wait and hope that it will come back to life, essentially. So this is a kind of typical day in the lab. And scientists have really not normal working days. It all depends on the experiment that we are doing. So one day you might have experiments until midnight, if you plan like that. Someday you might have half a day of experiments. So it really is depends on what you do and usually how you schedule your experiments. And I like this flexibility a lot, that I can determine what I do every day.
Same. Well, not now, because I'm not in science anymore. But yeah. So where was I? So going back to the Lamin blob, where would you start tackling the significance of it?
Well, so what I'm doing now is we have this collection of different genetic mutants of the genes that we know that somehow impact the death of the linker cell. So we do know that the death of the linker cell is programmed, which means that we know there's a kind of program of genes, one gene turns on the next gene, the next gene, the next gene, that leads to the death of the linker cell. And we know a lot of these genes that are involved in this. So I'm using some of these mutant worms in which the linker cells will abnormally survive. They will not die, they will abnormally survive. And I want to look at what happens to lamin in those cells. So that's one angle to what I'm doing. Another thing that I'm doing is I want to essentially decrease the amount of lamin in the linker cell. And I have this tool where I can do it specifically only in the linker cell. And it's called RNAi technology. And it's very useful in C. elegans when you want to decrease some gene. You just feed them the bacteria that codes for these short sequences that will match your gene of interest. And these short sequences will interfere with your gene of interest, and essentially you will have a decrease of your gene of interest. And so I'm curious to see if I decrease lamin in the linker cells, if that has an impact, if now the cells will not die, or what will happen to them. Yeah, so these are a couple of directions I'm pursuing.
So I know the period is ending. So for those who have to go, I think your teacher gave you permission to leave. For those who want to stay along, we can continue answering questions. But thank you, Olia. That was amazing. I was also fascinated by it. There was, are there more applications to the research you do other than cancer and progeria research?
Yeah, as I said, neurodegenerative disorders. Actually, we know one of the genes that directs linker cell death is very similar to the gene found in humans that is implicated in these neurodegenerative disorders like Huntington disease and other diseases like that. So it's a very direct and exciting link that we have. Overall, it's also similar to aging cells. So aging overall, these nuclear changes happen essentially in aging, and why do they happen? We don't really know. Yeah, so I would say cancer, neurodegeneration, cardiomyopathies. So these are different defects of the heart that lead to very early heart attacks, like in your 20s and your 30s. Muscular dystrophies, where your muscles just wither away. All of these kinds of diseases. So I think it's a quite wide spectrum of diseases. And one of the things I'm trying to determine is, which direction to go with my research, or should I do a little bit of that connects to different diseases.
And what got you into wanting to study cell death over other aspects of cell bio?
This kind of came a little bit from my PhD. My PhD was in this process called DNA methylation. So when you have your DNA and on the cytosines, you are able to add the methyl group. And for example, a big part of our DNA that used to be called junk DNA has this methylation mark on it. Also, a huge part of our DNA is composed of viral DNA, of viruses that infected us through all the ages and could incorporate their DNA into our DNA. This was a mechanism for the cell to tag this viral DNA, so the cell knows this is not my DNA. I cannot touch this DNA, and a way to silence this DNA. And there was just some interesting observation that when in these cells that are deficient for DNA methylation, that they were dying in a weird way, and not the way that we know, but a different kind of way. Also some of it was connected to different neuropathy diseases. And just kind of through this, I was curious to understand what happens with cell death. And I might go back to these deficient methylation cells to see what happens there. But I also really wanted, in my PhD, I worked with cells that grow in culture. And I really wanted to look at the actual organism. I did work with mice a little bit in my PhD, but I didn't enjoy it so much. A lot of people do it, but to me it was a bit sensitive. They were cute, and it was, hard to kill them. Doing dissections was fine, but to kill them was very hard for me. So I just wanted to work with some smaller organism that I could do research in. And I think that C. elegans worms are a beautiful model and easy to work with, and just being able to see the cell live and being able to see what happens to it live is quite amazing. So we can really observe this physiological process that unfolds, and it's probably a little bit less complicated than it is in mice or us. So once we figure that out, I do want to take it to mice in the future when I have my own lab, and but then people will be working for me, and they can work with mice and figure out what happens in mice.
So it's so funny because the session before, they preferred mice over monkeys, and you prefer worms over mice. So it's the hierarchy of like organisms that you work with. And then we'll just do one last question, and then we'll let everyone go. So now with coronavirus, do you have anyone that's helping or working with you?
Actually, we are not allowed to have new people, like new volunteers. I did have a high school student who worked with me last year, and I'm trying to hear back what are the rules of the university, if she can even come back to campus. Essentially, right now, some of the undergraduates who already had access to campus and were volunteering are able to come back, but new people are not allowed. That's why you guys, we are doing these virtual sessions with you. Otherwise, you would be coming here. Yeah, so unfortunately, because of that. So I don't have anybody working with me right now. I would love to in the future, after this all ends.
Same. Awesome. So thank you again. I don't know if there are any last questions for those who are hanging out. If not, we can dismiss, and hopefully we'll see you all in some of our other programs. Just a reminder for the SSRP program, if you all are interested, those applications are open and they close in December. Awesome.
Thank you so much, guys. Have a great day.
Bye.